Okay, and finally, BMAA, beta-methylamino L-alanine, and its toxicity. Moving on. It's the wrong place there. Yeah, we'll talk about where it comes from, its real-world impacts, and finish up by talking a little bit about spirulina. Okay, so what is it? It's a non-protein amino acid. It's found in cycads, which are non-flowering seed plants dating back to the Cretaceous or even Jurassic period, which I think I mentioned again later on. It's produced by Cyanobacterial, blue-green algae, associated with the root systems of these cycads. It may act as a chemical deterrent to herbivory, the consumption of plant material by animals, as usual. It's concentrated things like shark's fin, which people who eat shark's fin soup, well, I wouldn't say they deserve it, but moving on from that. Often you see algal blooms, which contain a lot of this and make seawater toxic. You hear of dogs dying if they've been for a swim in it. We're going to talk about fruit bats a little bit later on. There's an upside down fruit bat, and also have a look at Smarties and blue and spirulina. And there's the molecule drawn out. Algae are needed in many water bodies because they provide oxygen, are a food source for many other marine sources. In moderation, the algae provide balance for a healthy water ecosystem. However, due to reasons such as climate change, cyanobacteria can grow to excess, causing cyanobacterial blooms, which occur when there's enough sun, the water is warm, stagnant or slow moving, and if there are enough nutrients such as phosphorus or nitrogen present in the water body. So pollution and runoff from agriculture is a factor there. Blooms are more common during the late summer, early autumn. Current nutrient pollution is also exacerbating the problem, as I said, increasing the number of cyanobacter blooms that occur. And there's a notice by the sea saying don't go for a swim. Yeah, biomagnification, also called bioamplification or biological magnification, is increasing concentration of a substance such as a toxic chemical in the tissues of tolerant organisms at successfully higher levels in a food chain. There's a link there if you're interested for reading. And you see you go from the algae through plankton to small fish to big fish to sharks, and say which concentrated in parts of their tissue such as the fin. See this paper if you're interested for a review of the biomagnification effects of cyanobacterial neurotoxins among the Chamorro people of the island of Guam in the Pacific. Quite significant issues with this. This is where Guam is, say right in the middle of the Pacific, much fought over in World War II. Yeah, so this is the biomagnification process. While the amount in free cyanobacter and cygad roots is small, the seeds contain much more, and flying foxes that consume the seeds biomagnify the toxins further. And if you read the notes, there's more about cycads and the age of the cycads in the Jurassic period. Okay, so you may think, well, who cares about fruit bats? Well, bats sautéed in coconut milk is a local delicacy. Unfortunately, as we saw from the previous slide, it contains significant amounts of BCCA, which does appear to be associated with neurological issues in this particular population. Okay, spirulina, just to wrap up, I think. This is extracted from a blue-green algae, which isn't actually an algae in this case, but a filamentous bacterium. It's sold as a supplement, but it's also perhaps most widely known for replacing brilliant blue in blue Smarties. Not as blue as they were, but not as nasty as they were either. The issue of ingredients and their safety is an interesting one. Click on that link for more information. Unlike additives, they are not usually subject to rigorous safety testing, which is quite interesting. Algae as a food source of considerable interest. We've done some good projects here on this, and I will be offering one fairly soon. As well as other novel crops such as duckweed, which again we've grown here hydroponically. As often, however, there is a downside, as we've discussed. Blue-green algae products may be potentially unsafe. Again, you can read in the notes from Medline Plus there if you're interested, but it can contain a range of quite unpleasant things. A couple of definitions there. Microcystins or or ginosins, classes of toxins produced by certain freshwater cyanobacteria. So that's what he's mentioning there. Okay, so analysis, which you would think would be important, and it is, but it's difficult to do. This is one paper where people have done it. There's only been a few people have managed to do this successfully, and they've used quite sophisticated kit to do it. A few labs are actually qualified to carry it out, which may be something which becomes interesting when you think about the amount of spirulina that's sold. This is samples they used. They used raw spirulina from a range of samples, everything in triplicate, we'll notice. And they also used multi-ingredient drinks containing about 1.5 grams of spirulina per serving, and did this in a quartet of analyses. They didn't find BMAA in wool urine amounts, but the authors noticed some potential issues with the study, which are listed there. Added to these might be issues in quality control both at source and during the food chain. And there we are. That was the last food science and chemistry lecture. Phew.